Hello students, it is the second part of the chapter Principles of Inheritance and Variation. In the previous class we have learned about important terms and the terminologies used in genetics like variation, inheritance, monohybrid cross, recessive trait, dominant trait, etc. Now let us learn about test cross, laws of inheritance extracted from monohybrid cross, and some phenomena that are deviated from the law of dominance. Students, let's remember the phenotypic and genotypic ratios of monohybrid cross. From the cross we have done in last class, we know that phenotypic ratio is 3:1, means 3 are tall and 1 is a dwarf, and the genotypic ratio is 1:2:1, means out of the 3 tall plants, 1 is of capital T genotype and 2 are heterozygote tall, which are of capital T small t genotypes. Recessive trait dwarf is always homozygote, because a recessive trait like dwarfness cannot be expressed when the recessive allele like small t is with a dominant allele like capital T, means the recessive trait cannot be expressed when the genotype is a heterozygous condition, right? My point here is every individual that shows the dominant trait like tallness is not always homozygous, means all the tall pea plants we see are not always the genotype of capital T. Many of these tall pea plants may be heterozygote capital T small t. So we do not know the genotype of the individuals that show the dominant trait by observation only. To know the genotypes of F2 plants that show dominant traits, Mendel used a technique of genetic cross called test cross.
What is a test cross? It is a genetic cross between F2 plant or plants that show dominant trait with their recessive parents to determine the genotype of the plants.
Let us take an example. If you are given a pea plant having violet color flower, how will you know the genotype? Now your answer will be test cross, right? Here are the two conditions. The violet flower may be of homozygote capital W, or heterozygote capital W with small w. If it is a homozygote, gametes will be of one type by carrying capital W only, means 100% of the gametes produced by the homozygote plant will be same type, which carry capital W. So during test cross, the gametes carrying capital W will fuse with gametes of recessive parent carrying small w and produce only violet flower plants, right? If the violet flower plant is a heterozygote, 2 types of the gametes will be produced. Half of the gametes will carry capital W, other half of the gametes will carry small w, right? So 50% of the gametes will carry capital W, other 50% will carry small w. So after test cross, we will get 50% of the new plants will be violet, other 50% will be white. The phenotype and genotype of the test cross will be 1:1, means half of the new plants will be violet flower plants and other half will be white flower plants, right? So the phenotype will be 1:1, 1 violet, 1 white. Here let's know what is a back cross. It is a cross between F1 plants with one of their parents. So we know the test cross is a back cross with the recessive parent. Now let us try to know the laws of inheritance extracted from monohybrid cross. 2 laws which are extracted from monohybrid cross: law of dominance and the law of segregation. Law of dominance states the following statements: characters are controlled by discrete units called factors. Later they are known as genes. They occur in pairs. In a dissimilar pair of the factors, one member of the pair dominates the other. The factor which dominates the other is called dominant factor, and the other factor is recessive, which is not able to express whenever the dominant factor is present. Now, law of segregation. Law of segregation is a universal truth without exception. Law of segregation states that the factors or alleles of a pair segregate or separate from each other during gamete formation or gametogenesis in a way that a gamete receives only one factor or allele of the pair. Law of segregation can be understood easily from the monohybrid course we met earlier. Tallness is dominant and dwarfness is recessive. The F1 hybrid is tall. The recessive character dwarfness remains hidden in the F1 but reappear in the F2 generation without any sense. This is because the factor capital T and small t remain together in the F1 hybrid but segregated during gamete formation and enter different gametes. Half the gametes receive capital T, another half of the gametes receive small t. Diploid pair condition is restored during fertilization. Now let us talk about two phenomena that are deviated from Mendelism. All the patterns of inheritance could not be explained exclusively on the basis of Mendel's original principles alone, and certain complexes were observed by later workers which are deviated from Mendel's laws or Mendelism. One such phenomenon which is deviated from Mendelism is incomplete dominance. What is incomplete dominance? It is a phenomenon in which phenotype of the F1 hybrid offspring does not resemble any of the parent, but is intermediate between the expression of the two alleles in their homozygous state. Here, the phenotypic ratio is deviated from Mendel's monohybrid ratio, but the parental characters reappear in F2 generation. For example, in snapdragon flower Interinum and 4 o'clock plant Mirabilis jalapa, there are two types of flower color in pure state, red and white. On crossing the pure breeding red flower and white flower plants, hybrid of F1 generation possess pink flowers. And on selfing F1 plants, F2 generation comes as one red, two pink and one white. And also the genotypic ratio comes as 1 capital R, 2 capital R, small r, and 1 small r, small r. The phenotypic ratio and the genotypic ratio are same. Now we have codominance. It is the phenomenon in which offspring shows resemblance to both the parents. One such example is found in human ABO blood group. Suppose a father has blood group A, mother has blood group B. One of the possibilities of the blood group in their children will be AB blood group, right? So ABO blood group is an example of codominance. Both blood group A and B are expressed in AB type of the blood group. Now multiple allelism. Phenomenon of having more than two alleles for one gene is called multiple allelism. Example, human ABO blood group, because human ABO blood group gene is having three types of alleles: IA, IB, and I. Any individual who has blood group O is having homozygote II. So the expression of blood group is O. If individual is blood group A, the possible alleles may be homozygote IA or heterozygote IA small I. IA I, because IA is dominant over I. In the same way, individuals having blood group B are also having two possible pairs of alleles, IBI heterozygote or IB homozygote. Those who are having blood group AB, they will be always heterozygote of IA IB, right? Now, pleiotropy. It is a phenomenon in which a single gene product may produce multiple or more than one phenotypic effect. Example: a single gene that controls for the star synthesis in garden pea plants also control sizes of the seeds also. The alleles are given here: capital B, homozygote, small b, homozygote. If there is a cross, capital B, small b will be present in F1 plant. If the plant has capital B, the size of the seed will be largest and the amount of stars present here will be the maximum. And if the plant has a small b heterozygote, the size of the seed will be the smallest and the amount of the stars produced also will be the lowest. If the plant has capital B, small b, then size of the seed will be intermediate and the amount of the stars also will be intermediate. If we consider about the shape, capital B or capital B, small b will express as round shape. If the plant has small b, the shape will be wrinkle. Another example can be given as phenylketonuria, a disorder caused by mutation in the gene coding the enzyme phenylalanine hydrogenase. The affected individuals show hair and skin pigmentation and mental retardation. Okay students, let's do this much for today. In next class I will bring you dihybrid course and the laws extracted from this course. Okay.